head of the Department of Chemistry. He also is a very active member of the Polymers and Soft Matter group at MIT and the Broad Institute. So Jeremiah got his BS in biomedical engineering and chemistry from WashU, and his PhD in chemistry from Columbia. And there he did like very hardcore physical organic chemistry. He then moved to studying chemistry of materials in his postdoc with Dave Terrell and Bob Grubbs. And then he started his lab at MIT in, what, 2012? 2011, yep. Okay. And he basically invents new kinds of materials and new strategies to synthesize macromolecules with biomedical properties. And so I should also mention on the side, he does a little renewable energy storage and chemical sustainability. But we're focused on human health today, so Jeremiah's talk is the ABCs of antibody-bottlebrush conjugates.
All right. Thank you, Laura. All right. Thank you. So I'm going to start by saying it's really wonderful to be a part of this symposium and to have had the pleasure to co-organize it with Dane and to see many of my sort of heroes in this field present today. She's been unreal. So I'm super excited to tell you about some of the work going on in my lab over the years on what we call antibody-bottlebrush conjugates, which started out as more of a sort of synthetic curiosity, but I think hopefully can provide some new ways to think about targeted drug delivery moving forward. This project idea really came from thinking about the different ways we develop therapeutic windows with medicines. And here I show two, one sort of classical, I would say almost medicinal chemistry type of approach to develop small molecules that have a therapeutic window based on their mechanism of action, so selectivity for killing disease cell over healthy cell based on mechanism. And then a completely different way to achieve a therapeutic window is to bias where drug molecules go, i.e. drug delivery. And I mean, I think this symposium exists because antibody-drug conjugates, I would say, are the gold standard of that type of an approach. But you know what struck me now almost 15 years ago is the fact that we don't use these molecules in this context. Right? And in fact, today you see that there's a relatively small number of different payload molecules used in ADCs. And so a really simple question that we started to think about was, can we combine mechanism of action based and delivery-based therapeutic windows to generate more effective and safer cancer therapies? Just think about it very simply. If I could use payloads with a more selective mechanism of action, then every antibody-drug conjugate molecule carrying that payload to a target cell would deliver an inherently more potent drug to that cell, and every molecule that never reaches that cell would carry an inherently safer drug. That's the general idea. Now that seems pretty obvious, and you say, well, why don't we do that? And I think that's been captured in many of the discussions and the presentations from today, and sort of restate here from this nice review article published by Daiichi Sankyo a few years ago, where we know that for ADCs, the ones that have been successful have used highly potent payloads. And it just turns out that it's hard for medicinal chemists to make molecules that have sort of sub-single-digit nanomolar potency. We just don't have that many of those compounds. And so if we could enable using drugs that are 10 times or 100 times less potent, that could potentially open up a whole new landscape of the types of mechanisms of payloads we could use in ADCs. So how could we do that? If you want to use a payload that's 10 to 100-fold less potent, you could say, well, I could just dose 10 to 100 times more. We've learned from Greg, actually, sort of that in the clinic today, you know, all ADCs are given in the range of 1 to 10 milligrams per kilogram of antibody. And so if you were to say dose 10 times or 100 times more, that might get costly and challenging. So if you want to keep the same antibody dose, what's the only other way to do this, to sort of mash the payload toxicity or potency of existing payloads? That would be to increase the drug-to-antibody ratio by a factor of 10 or by a factor of 100. Right? And as we saw, actually, I think Carolyn had a figure like this in her presentation that, you know, if you try to do that and just put 10 times more drugs on an antibody, it's not going to work. You're going to disrupt the antibody function. And so you could think about trying to decouple where the payloads are attached, no longer to the antibody, but attach them onto some polymer chain that then gets attached onto the antibody. And this has been tried and developed, I think, most furthest advanced by Mersana, who we had a wonderful talk from earlier today. But this does face another issue, right? It's that if these are hydrophobic or somewhat hydrophobic payloads and you line a lot of them up onto a long chain, you're not going to really, you're going to reach a point where once this gets to a certain length, these are going to precipitate or they're going to aggregate. And so we can't just rely on this sort of design. So we started to think, well, how could we enable some sort of polymeric structure that could have a high density of drugs and linkers, but would maintain good physical properties, good solubility, avoid aggregation, with a wide range of different payloads and linkers. And that's where we proposed to install along each repeat unit of these polymers a hydrophilic side chain that extends just beyond the length of the payloads and linkers, shielding those linkers from exposure to the external environment, and therefore, in principle, giving us consistent PK, consistent physical properties, regardless of what the payloads and the linkers are. And you've seen this, I think, this sort of design sort of on the smaller monomeric or dimeric levels, also in Mersana's presentation and in Peter's presentation this morning. So if you imagine this now, this molecule, what it kind of looks like is a bottle brush, right? You might clean out a bottle with one of these things. And in fact, in the field of polymer physics and polymer science, that's what we call these molecules. What's distinct here is that we have these covalently attached drugs through cleavable linkers. So we refer to these as bottle brush prodrugs. And we think that they could enable novel ADC payloads and payload combinations. And so the question when we started the lab now, again, about 14 years ago, was how could we make molecules like this? We didn't have synthetic methods to make molecules like this, and as a chemist, that's actually what I like to think about, is making complex structures. Well, and if you think about this a little bit, you might realize that the most convergent, efficient way to make a molecule like this would be to synthesize a single monomer, a monomer that has a blue chain, a linker, and a payload, and somehow polymerize that. That way you would ensure that every repeat unit of your product has all the desired components, right? And in fact, that's the way we did try to tackle this challenge. And we've gone through several iterations of this, but the way we still make them today was a synthetic route developed by Hung Nguyen, a former graduate student in my laboratory. I won't walk you through the synthetic steps, but just briefly say that it's really efficient. We can make these on kilo scale with no chromatography. And this simple molecule is a norbornene imide that is subjectable to a polymerization reaction called ring opening metathesis polymerization, which I'll show you on the next slide. And it contains a 3 kilodalton peg chain and this terminal alkyne here. If you were here for the morning presentation, what do you think we're going to do with that alkyne? All right, so you're going to see. So next step, we do a little bit of medicinal-like chemistry. We take a small molecule payload of interest that maybe we want to use or a collaborator of ours is interested in using, and we design ways to synthesize prodrug variants. So we attach a cleavable linker with an azide, and in the presence of a little bit of copper, we're not doing this in a biological system, so we can use copper. We click this azide onto this alkyne, forming this, what we call a branched macromonomer, and now in a single polymerization step, by just controlling the stoichiometry of this monomer to a metal complex called the Grubbs third generation complex, we are able to form these bottle brush prodrugs. And using this general synthetic route, we have now used over 30 different kinds of payloads and imaging agents. We've done classic chemotherapies. We've done immunomodulators. We've done sting agonists, so similar compounds that we saw earlier. We've done PROTACs. I'll show you a little bit of work in that later today. All right. One thing I love about these, as I mentioned before, is that the hypothesis was that this unique architecture would make the physical properties, size, shape, solubility, sort of independent of what the payload and linkers are. So you could have this sort of modularity to change these variables without changing other factors. And here's just what I'm showing are 6 different BPDs with different payloads, bortezomib, pomalidomide, dexamethasone, a synergistic combination of those drugs, and an antagonistic combination of those drugs that we made just for comparisons, and a PEG bottle brush with no drugs. And they're all essentially identical in size by both size exclusion chromatography and dynamic light scattering. Here's just a cryo-electron microscopy image of one of those for your interest. And now the big question is how do you do this? So we're dealing with approximately 10 nanometer object and another approximately 10 nanometer object. We need to fuse them together in high yield, very selectively. Anybody want to guess how we're going to do that? Bioorthogonal chemistry, right? All right? And in fact, I added this, I hope I have time to talk about it, but I just have to mention another great thing about Carolyn. When I was a graduate student at Columbia, Carolyn came and gave a lecture. She was still, I think, junior faculty at the time. You were maybe on her tenure tour. They had just published their first JAX paper on the cyclooctine cycloaddition reaction with azides. And I went up at the end of the talk as a sort of second-year graduate student. I said, can I get some of those molecules? Those are really cool. And we published a paper together. There was actually the first use of this reaction in the sort of materials and polymer chemistry context. And I can just tell you, she didn't mention it, but this chemistry has also revolutionized that field as well. So just wanted to give a shout out. And now the reaction we used was not that one. It was a different one Carolyn showed. Uh-oh, what happened to my slide? Did I hit the black button? Don't hit the black button, apparently. Oh, what was that, the sound button? All right. There's a chalkboard behind this curtain. We know that if you've taught in this room before. Yeah, good job. All right, great. So long story short, you did see this reaction earlier from Carolyn. What we realized, and this was work done by Ben Liu, who is somewhere in the audience, right over there. He borrowed some chemistry from Will Gutekunst at Georgia Tech, where he showed that these ene-ine derivatives can terminate the ruthenium alkylidine chain ends of growing ROMP polymers very efficiently. So by simply adding this molecule 1 into this polymerization reaction, we can terminate these polymers, remove the ruthenium, and install exactly one tetrazine functionality onto the end of the chain that is then set up for that reaction that Carolyn showed earlier in the context of this click and release concept, where exposing this now to a monoclonal antibody that's been functionalized with a trans-cyclooctene, we conduct this inverse electron demand Diels-Alder reaction. But it's an extremely efficient coupling reaction to generate what we call the antibody-Bottlebrush conjugate. Now, in this paper, we've shown this both for the old school methods, so stochastic lysine functionalization. We've done site-specific cysteine conjugations, but I think it's pretty agnostic to however you would want to functionalize your antibody. So we can use this in almost any context. In this paper, because of the modularity of the system, we explored, I think, 6 different payloads. We used some of the classics like SN38 you've seen, which is also in Trodelvy, MMAE, which you've seen, but also things like paclitaxel and doxorubicin, which are not potent enough to be used in traditional ADCs. We also have played around with some PROTACs. We've used some fluorophores for imaging. And and here's a TEM image of one of these constructs, just showing you how uniform they are. And I failed to mention this, but one unique feature of the Bottlebrush architecture is that for a given molecular weight, they're about a third the hydrodynamic size as a linear polymer. So they're much, much more compact. So if you want to build in a lot of drug molecules into a small space, this is a very efficient way to do it without making your construct so huge, right? And that can be important for things like tumor penetration. We can synthesize these with different DARs. Here I'm showing a DAR 60 and a DAR of 120 run on a gel. If we do DAR 30, DAR 60, we don't see any substantial binding affinity decreases compared to trastuzumab and other antibody drug conjugates. If we go to 120, there's a little bit of attenuation of binding affinity, but it's still pretty good, and these can still function. And then just quickly, I'm running a little bit late, but I'll just fly through a few slides of some efficacy. This was our first study that Ben did looking at a DAR 135 SN38 trastuzumab conjugate compared to saline. The ABC gets essentially complete clearance of these tumors and 100% survival in this model. Notably, a Bottlebrush prodrug conjugated to IgG1 or the Bottlebrush prodrug by itself do display some efficacy, essentially as passive nanoparticle carriers, but not nearly as good as the targeted ABC. In head-to-head comparisons with TDXD, we see similar effects in high HER2-expressing cell lines. We do a little bit better in HER2-negative. I'm almost done. And then here's an example of doxorubicin, which again, not successfully used in the ADC context because it's about an order of magnitude less potent than typical ADC payloads. And here we can get essentially tumor clearance at clinically relevant doses of doxorubicin and antibody. And just in interest of time, we did basically the same kinds of results with PROTACs, and we can swap out what the antibody is. We used MUC1 in an ovarian cancer model. All right, so with that, I will finish and thank NIH, specifically NCI, for funding this work now for several years. More recently, American Cancer Society Yosemite, and also the Koch Institute for support. And lastly, I want to mention Window Therapeutics, which I am a co-founder of, and along with 3 of my former group members. Window is trying to bring these molecules closer to clinic and helping real patients. They also provided support for today's symposium, so thank you, Window. And I'm happy to take any questions.
Thanks, Jeremiah, and thanks for persisting. Do we have questions? Yeah, go for it, Peter. You're good at kicking us off.
Yeah.
Oh, yeah, PK. Can you talk about that?
Yeah, so these particles by themselves, have pretty long circulation half-lives, being essentially 10 nanometer PEG particles, like you would expect for such objects. And we essentially seem to maintain that in the context of the antibody conjugate. So the PK profile doesn't look so different than that of an antibody by itself, of what we've done so far. So.
Okay, this has to be short.
Very short. See, antibodies are perfectly very safe compounds.
Yeah.
And now we are loading with 200 or 300 unsafe compounds. So how do you are addressing the therapeutic window, the biggest question we have in ADC field?
Yeah, well, first note that the focus here is to have 100 unsafe compounds, but they're 10 times less unsafe than the ADC payloads that we normally see. So at the end of the day, that they're kind of equivalent, but you will have the same exact kinds of questions that we'd have to answer, right? The same therapeutic window questions would come up with any ADC payload. Maybe here, again, if we can use inherently safer compounds, that might provide an advantage, but I don't have enough data to say that we can do that in humans yet, at least.
The other question is,
Okay, no other question. Sorry,
Okay.
I'm very, very tough. I'm gonna hold everyone to it. So let's thank Jeremiah again.
Thank you.